Mate to Thrive Nation, I've got Martha Carlin on the show. She is an expert in gut health and gut bacteria. She's developed a lot of products, founder of BioCollective Lab, also the CEO of BioCollective, and a citizen scientist. Welcome to the show, Martha.
Thanks, Steve. I'm glad to be here.
Good. Now you're listening out there or you're watching on YouTube and you think you know a lot about the gut. We're going to start with some gut myth busters from Martha because she's been studying this for a long time. A real pain to purpose story. So we're going to get into a backstory. If you're listening out there and you've got a chronic disease, have you looked at gut, but not only your gut, the specific gut bacteria that you need to repopulate with and to destroy the bacteria that are possibly causing a lot of inflammation in your system. So your number one gut myth that people are believing out there, Martha. And if you can give us 3 or 5, just educate people that often think they know a lot because they know a little.
Well, people who have some sort of a chronic problem, they often don't connect it to their gut. But our gut is really that internal pharmacy that's making all of these critical things like neurotransmitters, hormones, our vitamins, all of that is made in the gut. And so if your gut's disrupted, it's very likely that that is ultimately the source of all your problems. And if you can address that, you're going to get to the root cause of what is causing your symptoms.
So maybe let me just summarize that. If you had a functional medicine doc or you had someone practitioner and you've got a chronic disease and they're not looking at your gut, there's a big possibility they're missing a big part of the progression of the disease.
That's absolutely true. And a big piece of that is they haven't been trained on how to look at it. We used to give people a 60-page report that had, all the genes, all the details, and they would take it into a doctor and their eyes would just kind of cross because they don't know what to do with that. Even a decade later, a lot of physicians still don't really know how to look at the microbiome and how to think about it. They think in terms of antibiotics and killing, and it's really an ecosystem problem, and it's very difficult to restore an ecosystem just by killing. We see that on our farms where, we kill specific weeds and we've exacerbated the problems we have in our soil. It's the same thing in the gut.
Okay, so number one myth is that you need to ensure that there's a gut link between yourself and your chronic disease, and you need to explore that and get to the right practitioner, correct?
Correct.
Okay, next myth that someone's listening out there saying, okay, I haven't done that, or what, I think I know something about the gut, but actually it's a myth.
Well, people think, okay, I need a probiotic. And they go to the store and it's a shelf full of probiotics. And they just think, okay, I can just pick anything off the shelf and that's going to solve my problems. There's a problem there in that probably about 90 to 95% of the probiotics on the shelf there that you're looking at come from just 3 suppliers. And there's roughly only 12 strains. Well, part of our problem is we've lost diversity, and you can't just throw a couple of strains together and solve a lost diversity problem. And so you need to look at the system and build the components of the system back with specific strains of bacteria and understanding, how they fit into that ecosystem of, are they producing vitamins? Are they producing neurotransmitters like GABA to help you be calm or sleep? So it's those specific things and understanding the need for specific strains that most people don't understand.
Okay, brilliant. Summary, it's very specific. The bacteria in your gut are very specific in terms of what they do and their function. And so if you're struggling in a certain area, now you're listening and you say, I struggle with anxiety, there's a chance that you need certain different bacteria or what a microbiome diversification to ensure that you can address that anxiety.
Absolutely.
Good. All right. Maybe one more, third myth in terms of someone listening out there.
Nutritional deficiency. So, in our modern world, our food supply is pretty nutrient poor. And, we think about the impact on our system of not having sufficient nutrition, but that actually gets worse down in the microbiome. So if you don't have enough nutrients for yourself, there's also not enough nutrients for them. So that makes the battle in your gut worse. So ensuring that you have appropriate nutrients for you and your microbiome, because we're actually, in terms of cells, 2 times more microbial cells than human cells. And in terms of genes, the microbiome is somewhere in the neighborhood of 100 to 300 times more genes in terms of our function. So all of those functions need nutrition. And so we need enough nutrition for us and also for our microbiome. And I would say a lot of people don't understand that means micronutrients, minerals are really essential for all the activity that's going on in there.
All right. So if you're listening out there, you're not feeding yourself only. You're a host to many bacteria or this microbiome. You got to feed them too because they're critical for your overall health. Is that a good summary, young Martha?
That is.
Brilliant. Okay, so 3 myths now. Let's talk about your backstory, how you got into this, because from what I understand, you're an accountant, you're a system sort of analyst. You obviously, have gone through this journey for a specific reason. And yeah, condolences for your husband that's passed recently.
Thank you. Yes, so my background was accounting, systems based. I was an auditor for a company called Arthur Anderson, which was the Big Eight, they used to call them, and really trained at how to look at a business to identify risk by mapping out everything that was flowing through the system. And I just carried that forward into my career. I became kind of systems operator and turnaround expert. And in 2002, my 44-year-old husband, who was a marathon running athlete, really good shape, never did anything bad, no drugs, no smoking, no nothing like that, was diagnosed with Parkinson's, an old person's disease. And I just kind of refused to take the standard dogma about that disease. And it just didn't make sense to me that somebody who was 44 would be getting an old person's disease. So I really started to dig in at that point. And of course, that systems-based approach, the first thing I looked at was food, because that's the main thing flowing through the system. And so I dramatically changed our diet, threw out all the processed foods. He had been drinking a soy protein shake every day, and I started to look at and investigate soy. And at that time, about 97% of the soy was GMO and likely had glyphosate residues, and started to understand those pieces. And just piece by piece, started studying the areas of science until in 2014, I read Dr. Martin Blazer's book. It's called Missing Microbes, and it's about the growing up in the age of antibiotics, which we have, and the connection to chronic disease. Because prior to that, no antibiotics, no chronic disease. We had acute disease. But these antibiotics have destroyed our microbiome because most of them are indiscriminate. And by doing that, that has altered this ecosystem and contributed to all these things. And I was like, that makes sense to me. It's like the gut is the general ledger of the body, and we've taken away a bunch of the assets. And so about 6 months later, the first paper was published where they showed they could divide the 2 types of Parkinson's. In Parkinson's, there's a tremor dominant and there's a posture and gait dominant. And they could separate people by the bacteria in their gut. And I was like, Eureka, that's it. I quit my job, started funding some research at the University of Chicago, where I met Dr. Jack Gilbert, who co-founded the BioCollective with me, along with Dr. Suzanne Vernon. And we started collecting people's poop samples, because your stool is a treasure trove of information, of genetic information, but it's also just looking at it. And this probably should have been in the myths. Like, can your stool tell you whether you're healthy? Well, It can. So in the European microbiome study, your stool quality and consistency was in the top 10. I think it was number 3 of the predictors of your long-term health.
Wow. Is there
And
a
we
study there,
found
or was that a study? Have you got a reference to that?
that was a study from the European Microbiome Project, and they
Okay.
looked at, I think it was like 200 factors or something.
Okay. Good. So we'll
I
put
could
that in the show notes, and you're listening up. Your stool and your poop is crucially important as a data point in terms of your health span and your lifespan.
And when I started the BioCollective and started collecting poop, one day I was out walking and this couple asked me what I did for a living, and I said, "Well, I'm kind of the poop queen." And I was explaining what I was doing, and they said, "Well, that makes perfect sense because that's how we know our dog is healthy. If we take our dog out every day, it's generally poops at the same time. If it's not going, if the consistency is wrong, all of that, that's how we know something's wrong with our dog." And so it's kind of odd that people know that about their dog and they don't think that about themselves. But in our laboratory, we actually found, so we collected whole stool. So a lot of people are familiar, if they do a stool test, they take a little
Yeah.
scoop and put it in a tube. We collected the whole thing, the whole shooting match, and homogenized it and divided it up into little tubes that we could share with other scientists. But in that process, the people in our lab who were processing those samples came to discover that they could identify a Parkinson's sample just by the stool. No information about the person at all, because people with Parkinson's have a stool that is the consistency of concrete. And you think, okay, well, concrete is a colloidal mass that no, water can't penetrate. Like if you have soil like that, it's very hard for nutrients to get in and it's the same kind of concept in the stool. And in Parkinson's, I think as we started to look, there's also, potential connections to lack of nutrient uptake. So back to that nutrition, there's loss of diversity in the gut, and it's shifted to, a microbiome profile that is producing hydrogen sulfide and methane, which is also connected with SIBO. So there's kind of all these pieces of the puzzle. But that was my journey into founding the BioCollective and building out this stool bank. And then we started to build computational models for how can we restore the gut. Like, you've got to put back these systems that work. And I happened to go to a Parkinson's conference where some researchers were talking about the sugar alcohol mannitol and its ability in an animal model to stop the aggregation of the proteins that contribute to Parkinson's and actually pull them out of the brain of the mouse. And so I came back and studied this little mannitol chemistry book, and I was like, wow, this is a really amazing molecule. And the first chapter actually had information about bacteria that ferment mannitol from glucose and fructose. And this was my kind of, well, we can put a system in the gut that can ferment mannitol, which will clean out the trash, and it will use up the excess in fructose and glucose that we have too much of in our Western diet. And so we made a little prototype for my husband, John, and within 30 days, he was no longer walking with a cane, feeling much better. And we were testing his microbiome all along the way and found that it was moving back closer and closer and closer to the healthy human microbiome profile. So that was back in 2017, and we really didn't decide to become a probiotics company until 2021, kind of after COVID. We decided that it wasn't too smart to be collecting whole fecal samples anymore, just from an exposure perspective. And so we brought the Sugar Shift probiotic to market and used our computational model and that systems thinking to build out a platform of products with specific targeted strains that actually work together as a team to produce the metabolites that you need for specific things, whether that's immune system or sleep or glucose metabolism or we have a new one now for anxiety called
Okay.
Perfect Peace. Yeah.
Okay, so that's the sort of backstory. And I mean, I know people are going to ask me what type, we had Dr. William Davis on the show, as I said. So if we go neurologically, we are going to put bioticquest.com and there's products there. But what are we talking about when it comes to neurological issues? If we're looking at Parkinson's, or a lot of people have got early onset dementia, what are the very specific strains that people should be looking at? I know they work symbiotically, but what are people looking for?
Well, so there is specifically another company besides mine that has actually done clinical trials in Parkinson's,
Yeah.
and there's actually is a single strain of Lactobacillus plantarum. Now ours is a system, and it has a Lactobacillus plantarum in it. That plantarum is it's a detoxifying microbe in
Mm.
many ways. So it can handle aluminum, which we're exposed to, so help remove aluminum. Our Lactobacillus plantarum breaks down glyphosate, which we believe is contributing to the degrading of the microbiome. So that's a strain to definitely look for. Dr. Davis talks about reuteri. I think reuteri is very beneficial too
Yeah.
because it produces those bacteriocins that address the pathogens in the small bowel, which is what Dr. Davis is focused on. We do have a reuteri in our sugar shift formula. And and that's an oxytocin producer. And there's dysregulation in the neurotransmitters in Parkinson's, and oxytocin kind of sits at the top of
Mm.
that cascade.
So
So I
does
think that
reuteri produce oxytocin, or are you saying plantarum? My understanding plantarum is very good for the immunity, but you're saying not only for the immunity, but for Parkinson's.
for Parkinson's, for detoxification.
Okay. So, and then the reuteri you're saying is for oxytocin.
Well, reuteri increases oxytocin production in
Mm.
the system.
Okay. And I mean, if people come, because 48% of our listeners on the US, if they come in and they're looking for something neurologically, because we do want to direct them to the right place. There's nothing sort of more frustrating for a lot of my clients and my community
Wow.
is if they go to Biotic Quest and they go to the shop, there's
You would go to the sugar
sugar
shift
shift.
probiotic. I
Yeah.
mean, the regulations are very strict about what you can and can't say, but
Mhm.
the sugar shift formula is the one I made for John, and that has the leuconostoc, reuteri, and bacillus subtilis, all three of which are the mannitol producers. So those are the ones that convert that glucose and fructose into mannitol, and that's also a free radical scavenger. So oxidative stress is another kind of
So the
driver.
sugar shift, you're saying for someone with a neurological issue, because I see reuteri, plantarum, how do you say this? Paracasi,
Paracasi, yeah.
paracasi, and then mesenteroides. So are those the ones that are for the neurological issues?
Well, it's a team designed
Okay.
to do detoxification, free radical scavenging. So again, it's not necessarily single strains. You're
Okay.
rebuilding a system that can help. What it does is it restores an anaerobic system in the gut. And a lot of the pathogens that we have issues with in neurological
Hmm.
problems, and frankly, other things as well, but is bacteria that thrive in an aerobic gut.
Okay. Good. And so, in terms of just shelf stabilization, I know there's a lot of sort of debate on this. Is this shelf stabilized? This lasts for a while if they ordered from you? Because I know that there are a lot of companies saying it has to be refrigerated. What's your view as sort of a gut expert there?
So we have tested ours shelf stable for up to 2 years.
Oh, wow. Sure.
I
Okay.
think there are some, I'll say, kind of hidden issues because
Mm,
we don't ship internationally
yeah.
anymore, but we did have some issues with people who were uh, trying to make yogurt
Okay.
when we shipped something to the UK, and we realized that oftentimes they're run through irradiation in international shipping.
Mm,
And I think that's an underappreciated contributor to shelf life problems. And the same thing would be with Amazon if you're not specifically. So now we do sell product on Amazon, but now that when we send them our inventory, we mark the box on all sides everywhere, do
okay.
not irradiate or X-ray. So that sort of exposure can certainly have an impact,
Mm,
but
okay.
and there are strains that are particularly heat sensitive. Um, Reuteri is one of those. I think above 106 degrees, Reuteri kind of dies off. But the other thing, maybe this should have been on the miss too,
Yeah.
that um, if your probiotics, like if they're dead, they have no action. And that is not accurate. So you have cell wall components or the debris of probiotic bacteria have immunomodulating effects. So you have the working living organism that is actively producing metabolism, but once it dies and its cell wall drops off, the cell wall components are what are called, some of them are called muramyl dipeptides. And we know there's a whole field of peptide therapy now. Those peptides have action in the body. So, and when you hear people talk about probiotics in terms of, oh, ours has pre, pro, and postbiotics, that's actually another myth because if you just think about when a strain is grown in fermentation, it's grown in prebiotics. And then you have living organisms, and they produce metabolites, which are postbiotics, and dead cell walls, which are postbiotics. And then it all gets freeze dried and you have pre, pro, and postbiotics in every strain of bacteria that goes
Wow.
into.
So you're not just taking the probiotic, you're taking the pre, pro, and the post because that's sort of metabolites. So you're getting it in. So it actually should write pre, pro, and post, shouldn't it? Because that's actually what you're
Right,
getting in there.
right.
Yeah. Okay.
So now the people who sort of pitch that they're the only people that have all three is a marketing ploy.
Okay. See, these are the things that people want to hear. These are the things that people don't know. So that's why, if I go into your website, if you look at the sugar shift, okay, now, I've gone on to the simple slumber. And I know we're going all over the place, but for someone battling with sleep, there's a change now. So have these been researched by your lab, or is this research studies that you can send me to say, okay, now we've got Lactobacillus acidophilus, and then I've never heard of this Leuconostoc mesenterides.
So
So
Leuconostoc mesenteroides is a microbe that is present in the vast majority, I won't say the vast majority, but a significant portion of fermented foods around the globe. It is the first microbe that grows in a fermentation like kimchi or sauerkraut or something like that. And it takes the oxygen out of the system and sets it up for the lactic acid bacteria to be able to do their job. So it's a sequence of steps that preserve that system. And Luke and Ostock is kind of the leader of the pack in setting up that. So it's if you want to get it and you can't buy the probiotic, like go buy some sauerkraut and
okay.
kimchi, and you will get Luke and Ostock.
Now, have you noticed that, say, for instance, with wearable technology like the Whoop and Aura, if you go and take this and there's Lactobacillus plantarum, Lactobacillus casei, and then Bifidobacterium longum and subtilis. So have you noticed a change in terms of the data on the wearable technology?
So we're actually in the process of recruiting about 60 people for two of our products to do
Mm.
some testing on that based on my son has a Whoop,
Mm.
and he's noticed, in fact, I don't know, I might be able to pull it up on my phone, but with our antibiotic antidote, improvement in his heart rate variability,
Oh, wow.
uh, like significant. And then the perfect piece, which doesn't go on sale until August 1st.
Mm.
But he sent me last week when he took his first dose of that, and you can see and it's within 30 minutes of taking it, everything just leveled out. It was kind of crazy.
Yeah,
So,
be good
um,
because there's a huge market out there with whoops or auras to have a look at what happens, with regards to, these probiotics. I mean, would you agree, just talking about the antibiotics, that you only take an antibiotic if you're in hospital in sort of a life-saving situation?
that's a that's a really tough question. And I'm going to go back to a story actually with my husband John. We went to Australia back at the end of 2009. So we already had Parkinson's. And we stayed with the people we went to visit there, had a place out in the countryside outside of Melbourne. And we went out there, and their water was a rainwater catchment. And when we got home, John became very lethargic, and he lost about 15 pounds in 2 weeks. And we started going through this
Mhm.
4 months of trying to figure out what was wrong, between scans and things showing up on the scans and blood work and blood cultures. No, they were unable to diagnose a specific infection, but he went in to get a bone marrow biopsy to see if he had leukemia. And the doctor said, well, while he's doing that, I think I'm going to try this really strong antibiotic. And I won't say that I would recommend this, but he gave him Levaquin, which that's a really bad antibiotic.
Yeah.
But he gave him that for 6 weeks, and everything turned around, and
Mm.
they never figured out what it was. And that was 2010. Well, fast forward to when I started collecting fecal samples, we think we figured out what it was, and it was a giant virus. They're called Mimi viruses. And we found it in his microbiome, and that particular virus was actually discovered 25 kilometers from where we were in
Mm.
Australia, which was odd. But you know what I would say, he wasn't in the hospital, but he was very, very sick,
Yeah.
and the antibiotics turned that around. And I do think he might have died if
Mm.
we hadn't
But
done
that's a
something.
life-saving situation. That's what I'm saying.
Yeah,
If you're not like,
yeah.
and that's what I tell my clients and my tribe is that you can't have it 3 days into a condition or 5 days or 7 days. You're really sick. You almost need to be hospitalized. You're not getting well. Your vital signs are failing. But what I'm just finding is that people in a modern day city are so busy, so rushed. And the quickest and the easiest way is they go to their medical doctor and they get a script for antibiotics. And I think it's destroying people's health. And so if you look at how important it is, I think a lot of these probiotics, including yourself, are so important because we're destroying our gut bacteria with overuse of antibiotics.
Absolutely. That is absolutely the
Mm.
case. And Dr. Blazer showed different classes of antibiotics have different impacts.
Mm.
And there's a lot of ecosystems research from the broader ecosystems. You can disturb an ecosystem and it comes back, but not exactly to the level that it did.
Mm.
You know, you have this kind of until you have a collapse in a system. And Dr. Blazer showed that some antibiotics the gut never recovers from.
Okay.
And the class of macrolide antibiotics, I believe, is one of the ones in that category.
All right. So,
And
yeah, I'll go for
Dr.
it.
Philip Shepperhans, who did the first Parkinson's microbiome study, later went on to, because I kept pressing him. I knew that people with Parkinson's, from talking to them, many of them had had a lot of childhood strep infections and taken a lot of antibiotics. And I confirmed that through some data with 23andMe that there was an increased risk if you had had 4 or more strep infections. And Dr. Shepperhans went into the Finnish national database because they had all those records, and they actually did show a correlation to increased risk of Parkinson's with a couple of classes of antibiotics within 10 years, like I think it was 3 or 4 courses, and then an antifungal within 5 years.
Sure. So someone like myself who hasn't had antibiotics in 27 years and really has had, when I say almost next to zero Western medicine, is there a probiotic protocol that one should follow? If I just look at, do they go very specific here? So for instance, if I go to your shop, the sugar shift is, obviously predisposed with my genetics and my family history to diabetes. Is there something that you'd want to take and then give yourself a break, or do microbial diversity programs just ensure you're not taking the same culture for too long? What are your thoughts here with regards to setting a plan in place?
Well, for somebody like you who hasn't had antibiotics in 27 years or something, and
Mm,
you're not exposed to Western medicine,
mm,
I would say, you could have, like for you, I would say the ideal immunity would be the
mhm.
best product to have as kind of a go-to
Okay.
if you're traveling places where you
Okay.
might get exposed, because it's very effective with foodborne pathogens, which everybody gets exposed to. And that would be a go-to product for you to just kind of have, but you don't have to take it all the time.
Okay.
And I would actually recommend somebody like you probably doesn't have to do the kind of gut repair or
Mm,
backup system that somebody else does. And fermented foods are really your best friend.
yeah. Okay, so I see here in this immunity, it's the Lactobacillus mesenteroides. Is that how you say it? Have I got it right now?
Yeah, mesenteroides. Yeah, mesenteroides.
Mesenteroides, and then the plantarum and the ruminus. Is that something specific now that I haven't seen?
So that is a specific strain that we actually isolated from a healthy human gut,
Okay.
and it it produces what are called these bacteriocins. So they're very specific, targeted weapons against pathogens. Our testing in the lab showed that it could eliminate 100% of listeria on the plate.
Wow.
So it's very effective against things like Listeria, Salmonella, E. coli, a lot of those pathogens that. And now we don't have this research on our strain, but I was looking at some research last month, and I'm thinking I might try to get similar done on our
Mm,
particular strain. But it was a ruminous, and it showed that it prevented, what part of its mechanism is preventing pathogens from being able to attach to your epithelial
wow.
cells.
I mean, this is very specific, and that's what I'm trying to get at is, Martha, is that you've obviously got the BioCollective lab. Tell us about that research, and you've obviously got your PhD there, because people I think are just taking, like you said, going to a store and taking just a probiotic. They're not sure if they're getting monoculturing, and they're just taking something general. I
Right.
think tell us about the lab and how you formulated all these formulas.
So the BioCollective started in 2015. We started collecting fecal samples and really looking at across the population, what do we see as the problems? And one of those was the loss of the anaerobic gut. But my chief scientific officer at the time was Dr. Raul Cano, and he was a microbial ecologist who spent 35, 40 years studying microbial ecology and using microbes to clean up the environment outside the body. He was a pioneer in using microbes to clean up oil spills. And so he was a very, very knowledgeable on kind of all the genetic toolkits of all the microbes. And so we sat down and spent a month kind of outlining, these are all the problems that we see that people have. How do we build teams that can restore, metabolic balance, target pathogens, restore a gut after antibiotics, We have ones for helping with female hormones that we haven't released yet, for supporting the production and conversion of vitamin A. All of these little kind of pieces of the puzzle, and then Raul, with his tremendous knowledge base, knew which microbes to kind of pull in to look at. And then we had to look at, okay, do they have GRAS status or not, which means they've been in food for a long time. And then we took our computational model, and this computational model, we call it BioFlux. And it's basically a flux balance model, which is kind of in an ecosystem. It is who's helping who out. How do they maintain balance in the ecosystem? One produces something and its waste is the food for another one. And so we built this computational model to be able to put a couple of strains together, or 6 strains or 8 strains, and say, over the course of 6, 8, 10, 12 hours, what are they going to do in combination as a team? Are we going to get production of the things that we want, like butyrate to feed the lining of the gut, or B vitamins, or bacterial melatonin, or tryptophan to help with sleep? So that's basically how we laid that out. Was the brilliance of Dr. Kano, or identifying the problems and then putting that into our computational model and proving out that his intuitive sense of which microbes do what together as a team actually worked.
Sure. Powerful, because you're making it very specific. And so there's no need for someone to go and do specific sort of GI map tests and Viome tests, or do you recommend that, or do they try your product first if they got anxiety?
It's a good question, because we used to test and give people information. And I do think it can be a valuable tool to understanding what some of the underlying factors may be. Do you have specific toxin producers? If you're working with a functional medicine person who knows how to look at something like that and kind of work with you. But oftentimes, people will look at a microbiome test and say, oh, like, we need to throw some antibiotics at this. And, that's going to make the problem worse. So I think intuitively, most people know what their primary drivers of their discomfort or symptoms, if that's what you want to call it, are. And they're looking for something to solve that. They've got severe constipation, or they've got anxiety, or they can't sleep. I mean, the people who can't sleep, it's kind of shocking to me, because I sleep really well. But can't sleep. Lots of people with anxiety, disrupted gut after antibiotics. I talked to a lot of people about that, or they had food poisoning and they just haven't really recovered. And so I think for the most part, people understand kind of what their symptoms are that they're trying to address, and they also quite often understand exactly when it started and probably what the driver was. And if you go into the kind of Western medicine pipeline, they don't ever ask you or really want to know necessarily what you think caused your problem, including Parkinson's, which back to anxiety and stress, traumatic events, stressors, and that sort of thing, I think is an initiator in Parkinson's. We've been doing a documentary on this. And what happens is you internalize that stress, it shifts the microbiome, and that whole gut-brain connection kind of starts to fall apart.
Okay, sounds good. I mean, your antibiotic antidote here, it's Bifidum, Subtilis, Acidophilus, Mesenteroides, again, Plantarum, Rhamnosus, Ruminus, and Thermophilus. Maybe just speak into that in terms of why that combination is important.
Well, so that product actually came about. It was kind of interesting. There was a study that came out of the Weizmann Institute in Israel, where they had studied an 11-strain probiotic formula for use after antibiotic treatment, and they actually found that it made things worse. And we looked at it, and we ran it through our model, and Raul and I sat down and talked about it. And he said, well, it's making the gut too acidic. So at that high acid level, you can't get a recolonization of a broader profile of bacteria. You need a system that is going to balance out the pH so that you can colonize with a broader spectrum of the microbiome. And so that's how we came up with our formula. Now, rhamnosus is a well-studied strain for immune modulation. Then, of course, we have the ruminous in there that I said, targets those pathogens. Often people took antibiotics because they had a pathogen. So you're just kind of maintaining that control, if you will. It's like, putting the guard at the gate to make sure the bad guys
Sure.
don't come back. And you've got that, the fermented foods logic is kind of how we think about it. Again, that the leuconostoc mesenteroides, which lays the foundation so that those other organisms can come in. And Streptococcus thermophilus is actually, that's one of the more well-known yogurt strains, and it's heat tolerant. So you have a mixture in there of microbes that are tolerant to different factors that helps balance out the system.
Sure, sounds good. Looks like you've done a lot of work here with regards to the very specifics. And I think probably the gap there is the specificity of these probiotics with regards to the condition. And you've spoken about mannitol. Just tell us about the bacteria that produce mannitol, why that's so important.
Well, what we haven't talked about is something called the glycocalyx. So I'll get back to that in a minute. But mannitol is a free radical scavenger. And so that's one of the really key things about it, is that it's picking up and cleaning up those free radicals. And back to the glycocalyx, that is what lines your gut. It lines your cells, it lines your cardiovascular system, it lines your brain. It's that sort of squishy gel. And it has an endothelial surface layer that is like seaweed. It's these stretched out sulfated polymers. And the interaction of that glycocalyx, it's kind of a molecular sieve that is protecting you from cellular debris, from various things. And those the little seaweeds are negatively charged, and mannitol is neutral. I'm probably going way too far down the science rabbit
No,
hole
no
here.
problem.
But the glycocalyx reacts very differently to different sugars, different salts, and different amino acids. And that's partly because of something that are called nanobubbles, and the bubbles are what are floating through that. So you have oxygen and nitrogen nanobubbles flowing through the glycocalyx and carbon dioxide coming out. So that's, a system of delivery for oxygen and nitrogen into the metabolism of the body. And mannitol just happens to be one of those sugars that is beneficial for the glycocalyx.
Okay.
And so when we go back to the original idea of where that came from, and the researchers in Israel who had showed it could help clean out the aggregates in the brain of the mouse, it's helping open up that barrier and pull out the trash.
Wow, well said. That's incredible in terms of mannitol and the bacteria that produce the different parts from, these neurotransmitters, GABA. It's very specific. So maybe you can just speak into someone's listening and saying, okay, where do I start? Do I go get a general probiotic? Do I have to change my probiotics? Should I do a spore biotic? How do I take the simple steps with regards to probiotics?
Sure. So, spore biotics, they're also interesting. You probably don't want to overdo them, but we have a spore biotic that is in every one of our formulas. So we have DE111, which is Bacillus subtilis, which is in all of our formulas. But we're actually just coming out with a gut repair kit that is kind of a three-step process where you would start with the antibiotic antidote. And most people say, okay, well, I haven't taken any recent antibiotics, but this should have been on the myths too. Most people think, oh, I haven't I haven't taken any antibiotics, but you have, because most of the food preservatives in the prepackaged food at the grocery store are antibiotic because they don't want microbes
Yeah.
growing on the food, and they want to extend the shelf life. And so there are many antibiotic ingredients in processed foods or even the fresh packaged foods. So you have been exposed to antibiotics. And then, of course, there's antibiotics in the animal supply because they're used in cattle and chickens and
Mm.
quite frequently. So it's the antibiotic antidote. You do that for a month. Then you do the ideal immunity to make sure you're kicking out any bad guys that were there. And then we move you on to sugar shift, which is that great overall metabolic health probiotic that kind
Mm.
of balances out glucose and improves energy, produces butyrate to feed the lining of the gut, restores that anaerobic gut environment. And so, that's a stepped process we've put in place because people have been asking for that. Okay, how do I do this? What do I do?
Mm. Great. Well done to you. Maybe you can just sort of give a message, hope with gut health. Looking at 26 years of practice, I've never seen people so sick, from autoimmune to anxiety to depression to despair. I think a big part of it is the gut, but it's obviously linked to the vagus nerve, the stress that we're under, the radiation from the electromagnetic fields affecting our gut and our nervous system. I think, how do you see the future with regards to gut health and chronic disease?
Well, I am hopeful, but it is a challenge. I will back to you had someone on a few weeks ago who was talking
Mm.
about Crohn's and colitis and the incidence of that in the United States and Canada. And I'm actually working with a Dr. Tom Dow, who has been a pioneer in looking at autoimmune after retiring from a career in eye surgery, um, and connecting it to Mycobacterium avium paratuberculosis, which is endemic in the milk supply. And they also found that, like, people in Bahrain did not have Crohn's and colitis until cattle from a country, I don't know if it was America or somewhere else,
Mm.
but a country where the Mycobacterium avium paratuberculosis was endemic in the cows, until those cows were shipped to Bahrain. And now in Bahrain, they have Crohn's and IBS and colitis. And so Tom has been a pioneer in this and has a specific strain of bacteria that they have studied in the cows and in humans. And we're actually helping him to produce that. So we're in the process of prototyping our scaled up growth for the strain is called Dietzia, and it's a pretty interesting and remarkable strain. And so I am hopeful for that because, and he has two clinical trials on the horizon waiting for us to get the scaled production ready. But I think we're starting to see how some of these drivers that are coming from the food supply into our guts and how we might be able to address that. And I think you'll start to see more and more solutions as the understanding gets deeper about what those drivers are. And then that glycocalyx, which maybe we can talk about that another time, but I think that's going to be a huge paradigm shift. And I would encourage you to look up Dr. Hans Vink, who has spent 35 years studying the glycocalyx, and supporting that is going to support our gut health and our overall health.
Brilliant. Well, I want to declare favor and blessing over you, Martha, that you would rise up in this amazing calling. I know it's been a painful journey, but it's been a journey of discovery to help others, and that's the service that you're giving. And so I know there's been an incredible sort of connection because I think you're really in it for the long haul. Not, yes, it's a business, but it's to change people's lives. And so we salute you, we honor you, and yeah, let's chat offline because obviously you got IP that you possibly could license out there. But thank you so much.
Thank you.